Cultured beef meat, also known as lab-grown or cell-based meat, is produced without slaughtering animals. Instead of raising cows for years, factories grow real beef meat directly from animal cells inside highly controlled industrial environments. This revolutionary process combines biotechnology, food science, and advanced factory systems to create meat that looks, tastes, and feels like traditional beef. Inside a cultured beef factory, everything is designed for precision, cleanliness, and biological safety. The entire production takes place in sterile chambers that resemble pharmaceutical labs more than traditional slaughterhouses. Every step is monitored by computers, scientists, and automated machines to ensure consistency and food safety. The process begins with cell collection, which is the foundation of cultured meat production. Animal cell selection. The factory starts by selecting a healthy cow with strong muscle development. A small, painless biopsy is taken from the animal, usually from muscle tissue. This biopsy is about the size of a grain of rice and does not harm the animal. From this tiny sample, millions of cells can eventually be grown. The collected tissue is immediately transferred into sterile containers and transported to the laboratory section of the factory. Temperature control is critical during transport to keep the cells alive and stable. Isolation of muscle stem cells. Once inside the lab, technicians isolate specific cells called muscle stem cells or satellite cells. These cells are special because they can multiply and transform into muscle fibers, which make up beef meat. The tissue sample is carefully broken down using enzymatic solutions. This separates the muscle stem cells from fat, connective tissue, and other unwanted materials. Advanced filtration systems ensure that only the strongest and healthiest cells are selected for growth. Cell health testing and quality control. Before any growth begins, the cells go through rigorous testing. Scientists examine them under high-resolution microscopes to check their structure, growth potential, and genetic stability. Any weak or damaged cells are discarded immediately. The selected cells are then stored in nutrient-rich solutions that keep them alive while preparations are made for the next stage. Strict hygiene protocols are followed, including air filtration, sterilized equipment, and protective clothing for all workers. At this stage, the factory has successfully prepared the biological building blocks of cultured beef. From a microscopic sample, the foundation for large-scale meat production is now ready. After the muscle stem cells are isolated and approved through quality control, the factory moves into the most critical phase of cultured beef production, cell nourishment and rapid expansion. This stage determines how efficiently and safely meat can be grown at an industrial scale. Preparation of the growth medium. Inside a separate sterile processing area, the factory prepares a special liquid called the growth medium. This medium acts like artificial blood for the cells, supplying everything they need to survive and multiply. The growth medium contains purified water, amino acids, glucose, vitamins, minerals, salts, and carefully balanced growth factors. These ingredients are mixed inside stainless steel tanks equipped with temperature controls and automated stirrers. Sensors continuously monitor pH levels, oxygen content, and nutrient concentration. Every batch of growth medium is tested for contamination before use. Even a small impurity can destroy an entire production cycle, so the factory follows pharmaceutical-grade safety standards. Seeding cells into bioreactors. Once the growth medium is ready, the selected muscle stem cells are gently introduced into large vessels called bioreactors. These bioreactors are the heart of the cultured beef factory. They can range from small laboratory-sized units to massive industrial tanks holding thousands of liters. Inside the bioreactor, conditions mimic the natural environment inside a cow's body. The temperature is kept warm, oxygen is carefully controlled, and nutrients flow constantly around the cells. Slow, continuous stirring prevents the cells from clumping together while avoiding damage. Rapid cell multiplication. As time passes, the cells begin to divide rapidly. One cell becomes two, two become four, and soon millions of cells are growing simultaneously. Automated systems track cell density in real time and adjust nutrient supply accordingly. Waste materials produced by the cells are filtered out, while fresh nutrients are added without stopping the process. This continuous flow system allows cells to grow efficiently and consistently. Scaling up production. When cell density reaches optimal levels, a portion of the cells is transferred into larger bioreactors. This scaling up process allows the factory to move from millions of cells to billions, all while maintaining uniform quality. Robotic arms and sealed pipelines handle transfers to avoid human contact and contamination. Every movement is logged digitally, creating a complete production record for safety and traceability. By the end of this stage, the factory has grown enough living cells to begin forming actual meat structure. What started as a tiny biopsy has now become a massive volume of living beef cells, ready for the next transformation. After massive cell expansion inside bioreactors, the factory shifts focus from growth to structure creation. At this stage, cells are no longer just multiplying. They are guided to become actual muscle fibers and fat cells, which together form real beef meat. Triggering cell differentiation. To start differentiation, technicians gradually change the composition of the growth medium. Certain growth factors are reduced, while others are added to signal the cells that it is time to stop multiplying and start transforming. Temperature, oxygen levels, and nutrient flow are adjusted with extreme precision. These changes tell muscle stem cells to stretch, align, and fuse together, forming long muscle fibers similar to those found in natural beef. Formation of muscle fibers. Inside specialized differentiation bioreactors, cells begin aligning themselves in parallel structures. Gentle mechanical stimulation is applied using micro-movements inside the tanks. This stimulation mimics natural muscle movement inside a living animal and helps create stronger, denser fibers. Over time, thin muscle strands grow thicker and longer. The factory uses microscopic imaging and AI-based monitoring to ensure that fiber thickness, length, and density meet quality standards. Fat cell cultivation. At the same time, a separate batch of stem cells is encouraged to become fat cells. These fat cells are essential for flavor, juiciness, and aroma. Without fat, cultured beef would taste dry and bland. The fat cells are grown in their own controlled environment with a different nutrient balance. Once matured, they are carefully combined with the muscle fibers in precise ratios depending on the desired cut of meat. Three-dimensional meat structuring. To turn loose fibers into recognizable meat, the factory uses edible scaffolds made from plant-based or biodegradable materials. These scaffolds provide support, allowing muscle and fat cells to grow in 3 dimensions instead of flat layers. Cells attach themselves to the scaffold and continue growing, slowly filling the structure. Over time, the scaffold either dissolves naturally or becomes part of the final edible product. Texture and quality enhancement. During this phase, electrical or mechanical stimulation may be applied to improve muscle firmness and chewiness. This process helps replicate the texture of traditional beef. Sensors constantly monitor moisture content, protein density, and fat distribution. Any batch that does not meet strict quality standards is removed from the production line. By the end of this stage, the cultured meat now looks and feels like raw beef. It has muscle fibers, fat marbling, and structure, all created without slaughter. Once the muscle fibers and fat cells have fully matured and formed stable 3-dimensional tissue, the cultured beef is ready for harvesting. This stage transforms biological tissue into recognizable beef cuts through precise industrial methods. Harvesting the cultured meat. The bioreactors are gradually depressurized, and the nutrient flow is stopped. Automated systems gently separate the grown meat tissue from the growth environment without damaging the fibers. Robotic tools lift the meat structures and transfer them to harvesting tables inside sterile chambers. At this point, the meat resembles raw beef but has never been exposed to open air or human touch. Initial cleaning and moisture balancing. The harvested meat is rinsed using food-grade saline solutions to remove any remaining growth medium. Moisture levels are carefully balanced to achieve the correct juiciness and firmness. Excess liquid is removed using gentle vacuum systems that prevent fiber collapse. This step ensures uniform texture throughout the meat. Shaping into beef cuts. The cultured meat is then shaped into different cuts such as minced beef, patties, strips, or steak-like portions. Precision cutters and molding machines shape the tissue based on product requirements. For thicker cuts, multiple layers of cultured meat are combined and pressed together under controlled pressure. Fat distribution is adjusted to mimic traditional marbling patterns. Controlled aging process. To develop deeper flavor, the meat undergoes a controlled aging process. It is stored in temperature and humidity-controlled chambers where natural enzymatic activity enhances tenderness and taste. Unlike traditional dry aging, this process is faster and more predictable. Sensors track protein breakdown, aroma development, and texture changes in real time. Flavor enhancement and final conditioning. Natural flavor compounds may be introduced to enhance beefy aroma and richness. These compounds are derived from yeast fermentation or plant sources and are carefully balanced to avoid artificial taste. The meat is then rested to allow flavors to stabilize. During this time, texture firms up and moisture redistributes evenly across the tissue. At the end of this stage, the cultured beef is fully formed, flavorful, and structurally identical to conventional beef. It is now ready for safety checks and packaging. After harvesting, shaping, and flavor development, cultured beef enters the most strictly regulated phase of the factory. This stage ensures the meat is safe, stable, traceable, and ready for consumers around the world. Comprehensive safety and quality testing. Before packaging, every batch of cultured beef undergoes multiple safety tests. Samples are taken to on-site laboratories where microbiological, chemical, and structural analyses are performed. Technicians test for bacterial contamination, toxins, abnormal cell behavior, and nutrient balance. Advanced DNA screening confirms the cells remain stable and unchanged throughout the process. Texture, color, aroma, and moisture levels are also inspected. If even one parameter falls outside approved limits, the entire batch is rejected and safely disposed of. Only meat that passes every test moves forward. Final product classification. Once approved, the meat is categorized based on product type: minced beef, burger patties, steak-style cuts, strips, or ready-to-cook portions are separated into dedicated production lines. Each product type has its own packaging format, weight standard, and storage requirement. Automated systems ensure accuracy so every package matches its label exactly. Vacuum sealing and modified atmosphere packaging. Packaging takes place in sterile, climate-controlled rooms. The cultured beef is placed into food-grade trays or pouches and sealed using vacuum technology or modified atmosphere packaging. By removing oxygen or replacing it with protective gases, the factory extends shelf life without preservatives. This method also locks in freshness, color, and flavor. Packaging machines work at high speed, sealing thousands of units per hour while maintaining pharmaceutical-level hygiene standards. Labeling and traceability systems. Each package receives a detailed label containing production batch numbers, nutritional information, storage instructions, and regulatory approvals. A digital QR code links the product to its entire production history. From the original cell sample to the final package, every step is recorded. This full traceability allows instant recalls if needed and builds consumer trust. Cold storage and inventory management. Packaged cultured beef is moved into cold storage rooms where temperature and humidity are tightly controlled. Smart inventory systems track stock levels and expiration dates in real time. Robotic systems organize products for shipment, ensuring first-produced batches are shipped first to maintain freshness. Distribution to global markets. Finally, the cultured beef leaves the factory through refrigerated transport. Cold chain logistics keep the meat at safe temperatures during transport to supermarkets, restaurants, and food distributors. Unlike traditional meat supply chains, cultured beef factories can be built closer to cities, reducing transportation time, emissions, and spoilage. By the time the product reaches consumers, it is real beef meat grown in a factory, identical in protein and taste, but produced with advanced technology, precision control, and no slaughter.